Yes. So what I have done is I have just switched on my screen recording so that whatever I'm presenting is recorded for you guys, and I'll upload it on a YouTube channel, the link of which will be shared to you guys in case someone needs to refer it uh, whenever they want. And these slides that I'm presenting as material will be also provided to you in a Google Drive link. It is better for all of us who are attending this course to attend these live sessions to gain the maximum benefit. Let me share my screen now that I have. Okay. Yes. So can everyone see my shared screen? Just let me know in the chat box with a yes or a like, and we can move forward. Very good. And my voice is clear enough, I guess so. So once again, welcome everyone to the first class of Environmental Biotechnology, week 1. And this is taught by Professor Pinaki Sar from IIT Kharagpur. And my name is Ashutosh, and I'm a PhD scholar at IIT Kanpur. So I'm appointed by the PMRF, which is the MHRD ministry driven scholarship programs, which offers PhD students opportunities to teach and assist professors in NPTEL courses as well. So this is the format. I will repeat the format again for everyone, as well as let me type it in the chat box. So the format would be similar to what you will face in your assignments. These are multiple choice questions with one option correct. And these are some of the example questions, not the exact questions which you will be facing in your assignment. And we will try to solve these questions one by one and understand the background and explanation behind it, in a way recapping the whole week lecture. So we will start with a general introduction of this course, which anyway Professor Pinaki has given for lecture week 1. So this is the first question on your screen. Which of the following statements is incorrect? So this is a simple question. There are 5 statements and you have to tell me which of the following statements is incorrect. Option A, environment includes both living and non-living things. Option B, biotechnology uses microbial, plant and animal cells and their products. Option C, mitochondria and chloroplast resemble prokaryotic cells. Option D, fermentation technology is a new invention arising after the establishment of germ theory. And option E, microbial biocatalysis are generally cheap and environment friendly. So there are these 5 statements, and you have to tell me which of these following statement is incorrect. Only one option is correct. A, B, C, D or E. Let me know in the chat box which option do you think is the correct answer. And for all those students who have joined recently, welcome to this live TA session for Environmental Biotechnology. We will be having these classes on Saturday, every Saturday, 3 p.m. to 5 p.m. unless prior notice. And this will be an interactive session where I will pose certain questions related to the current week, lecture week, and we will solve some questions. Interact with me via the chat box, and let's move forward. This is a recorded session, and the recordings will be available on YouTube. Okay, so let's take a minute and check what have people answered in the chat box. I already see many answers. Nandini has answered D, Tapsi D, Tuhin, Vishwa, Vidhi have all answered option D. Manal, Utkarsh, Snehal, Yashashvi D. Okay, Saida has also answered D. Very well. Good, good, good. Keep up this enthusiasm while we reach till the last of the session. We have ample of time, and 10 to 15 questions should not take much time. You can also stall me anytime if you do not understand the explanation, and I'll be happy to go through it again. If I'm not able to answer any question, do not worry. In the next class, I will find the answer out and try to solve your doubts. Okay? So which of the following statements is incorrect? The correct answer to question number 1 is D. Fermentation technology is a new invention arising after the establishment of germ theory. So congratulations to all the students. Everyone has answered correctly. Option D is the incorrect statement. So option A is correct because environment includes both living and non-living things, while fermentation technology is not a new invention. Fermentation technology is an old invention from dating back to 7000 BCs or even before that. And germ theory is fairly recent, coming from the 1500s. So we will see what all these options are. So the definition of environment itself says that environment encompasses all the living and non-living elements that interact and influence life. So it has a living component. For an example, if the Earth's environment, for the Earth's environment we have the living components, which are plants, consumers, animals as humans and other animals, and microbes, which are the decomposers, while there are abiotic components like light, temperature, gases and water, right? So both these in abiotic and biotic components, the living and non-living elements, make up and interact together to make what is known as the environment. So option A is not the incorrect statement. Environment includes both living and non-living things. Now coming on to why biotechnology uses all these forms of organisms, microbial, plant and animal cells and their products, all to put to their use as biotechnology for enhancing the quality of the environment. Looking at option C, mitochondria and chloroplast. If we look at the history of the mitochondria and the chloroplast, there is a theory of endosymbiosis which says that organelles which are double membraned, so in an animal cell which is a single membraned cell, we have only one membrane, which is the plasma membrane. While if you look at primitive organisms like bacteria and the prokaryotes, they are double membraned organisms. So similarly, we found some organelles which are subcellular in nature, like mitochondria in the animal cells and chloroplast in the plant cell, which are also double membrane, and they also surprisingly have their own DNA. Did you guys know that the mitochondria as well as the chloroplast have their own DNA other than the cellular DNA? So there is a theory that evolved which says that these organisms entered into a symbiosis relationship. Symbiosis means a relationship in which both the organisms benefit from each other, and live together. Symbiosis means living together. So the mitochondria, which was earlier a prokaryote, in the young earth there supported many types of bacteria. The prokaryotes were the first kind of life to originate, and most membrane enclosed organelles, including the nucleus, the endoplasmic reticulum and Golgi, originated from deep folds in the plasma membrane. So this talks about the story of how life originated on Earth. And then comes this theory that mitochondria and chloroplast originated as bacterial cells that came in to live inside the larger cells. So they formed an environment and they got engulfed by a larger eukaryotic cell. And this is the theory of endosymbiosis. How a double membraned organelle which has its own DNA, which has its own genetic material like a mitochondria or a chloroplast, came inside, got engulfed by another larger eukaryote and had a co-living relationship like partners. They lived in endosymbiosis. Mitochondria and chloroplasts are therefore considered to have originated from bacterial cells or prokaryotic cells, specifically bacteria that were engulfed by early eukaryotic cells in a symbiotic relationship. So that is why option C is also a correct option. Then going on to the option E, which is the last option, microbial biocatalysts. They are very cheap. The last option, if you remember what was the last option, I'll show the question again. Microbial biocatalysts are generally cheap and environment friendly. This is absolutely correct because there have been numerous examples in which such microbial biocatalysts have been used in many applications like synthetics of biopolymer, creating flavors and fragrances, the fermentation industry, like beer and curd and cheese used as biofuel or in medicinal use, right? So these catalysts are microbes and enzymes derived from or enzymes derived from the microbes which are used to accelerate or catalyze chemical reactions, often in industrial settings, and they are relatively cheaper. That is why they are being used in upscale and large scale industrial processes. And let us come to the only correct option, which is option D. How old is this fermentation technology? So this is an image showing Egyptian pictures dating back so early as 7000 BCE with evidence of fermented beverages consumed in places like China, Mesopotamia, and the ancient Egypt. It was developed empirically long before the establishment of germ theory. Now, what is germ theory? Germ theory is something which is coined by Louis Pasteur and Robert Koch, and these are the two great scientists who gave the germ theory. While fermentation has appeared way before than germ theory. Okay, there have been evidences in ancient civilizations about fermented food like beer and fermentation of milk and bacteria. So option D was the correct answer, which all the students have answered correctly. Well done. Is it clear to the students? Just let me know in the chat box and we will move forward. I hope it was a pretty easy question. Very good. Thanks for the response. I see many responses. Thank you. Okay. Moving on to the second question of this evening. Question number 2. What are the key goals of environmental biotechnology, and which of these options does not include the key goals of environmental biotechnology? Right? Another correct, incorrect choice type question. Which of these options, key goals of environmental biotechnology, does not include which of these options? I hope everyone can see my screen, see the question clearly. I'm also reading it aloud for all those who want to listen to it. Option A, replacing plant and animal products with microbial alternatives. Option B, prevention of environmental pollution. Option C, generating valuable resources for human society. Option D, cleaning up of contaminated environment. And option E, designing sustainable alternatives to fossil fuels. I think this is also an easy question for you guys. What are the goals of environmental biotechnology, and which of these does not align with the key goals of environmental biotechnology? Okay, let us wait for a minute for everyone to answer. I think, what is the strength of this class? Can I see? Meanwhile, I see many people are also already answering. I think 16 to 17 people. Okay, the strength is small, but I want everyone to answer because these are pretty easy questions, and the more you participate with me in this session, this will be fruitful for you guys, because all the lectures in the NPTEL are all recorded, pre-recorded. You can go through a lectures, but this is the only live session that you can benefit from in your whole course. Okay, I see many answers coming up in the chat. Nandini and Tuhin and Snehal have answered A. Saida A. Manal is saying option D. Utkarsh A. Vishwa says C. Krishanu, Yashashvi, Akshat A. Tapsi says C. And Ankita is saying A. Okay, there's a lot of mixed opinions. Many people say A, some are saying C, and people are also confused on D. Okay. I think most of the people have answered, and let us see what is the correct answer to this question. Question number 2, key goals of environmental biotechnology. Mind the spelling mistake, it is environmental biotechnology. Does not include which of these options? The correct answer is option A. replacing plant and animal products with microbial alternatives. Right? Option A is the correct answer. So, to all those who have answered option A, congratulations. People who could not get it right this time, you will have another chance in all these next questions. Okay? Do not need to have a small heart. Option A is not one of the key goals of environmental biotechnology. So, see, these options are designed as such that they can be confusing at times. Now, if I let's say you that option A is actually more industry oriented, in which in industrial biotechnology we want to replace plant and animal products with microbial alternatives, because microbe have several advantage in an industrial setup, right? Because of their small size and rapid generation time, they double in hours, not like plant, they reproduce quickly, and they provide also higher surface area. So these are all points which are covered, which are advantageous in industrial biotechnology application, but does not align with one of the key goals of environmental biotechnology. Okay, now let us look at the other options which are actually the goals of environmental biotechnology. What does these goals say? Prevention of environmental pollution. One of the important goals of environmental biotechnology is to use biological organisms, is to use the power of biology to prevent environmental pollution, to reduce pollution. How? Techniques like bioremediation use microorganisms to break down pollutants, as shown in the first image where a scientist examines microbial cultures. So here we see like technique like bioremediation. Bioremediation, we introduce a microorganism to improve the quality of the environment. Suppose a water body, we introduce, suppose there is an oil spillage or there is a plastic infestation in a water body, we use oil digesting microbes like fungus and bacteria which can digest oil to reduce an oil spillage. So this is how is a good example of prevention of environmental pollution using bioremediation. Generating valuable resources for the human society. This is the second goal of environmental biotechnology. How to generate resources which can be utilized further for the human society for our own benefit using microbes and using biotechnology. This involves producing biofuels and biodegradable materials, and this can be also as used in future for human consumption, right? We all know how the current era is dependent on the use of fossil fuels, and we are running out of reserves of fossil fuels, right? So involving biotechnology, involving microorganisms and other organisms for producing safer and alternatives and resources for human consumption, like for biofuels and materials which are easily biodegradable and pose less harm to the environment, right? Then what is the next objective or key goal of environmental biotechnology? Cleaning up of contaminated environment. It is also very similar to what we saw in the prevention of environmental pollution. An environment which is already contaminated, how can we clean it with the help of environmental biotechnology? To detoxify polluted sites and the practical application of these, like using wastewater management and sewage treatment plants, and designing sustainable alternatives of fossil fuels also aligns with generating valuable resources for human society. So these are the four options that we saw in our earlier question. Option B, C, D, E are mostly aligning with the key goals of environmental biotechnology. And what is with option A? Replacing plant and animal products with microbial alternatives is not a primary goal as such, because environmental biotechnology prioritizes restoration of normal ecology and sustainability over product substitution. So you need to understand the basic difference between an industrial biotechnology and environmental biotechnology. Environmental biotechnology prioritizes ecological restoration, like the restoring the environment and how sustainable it is, while industrial biotechnology maximizes the product profit, generating more amount of product and resources with less amount of input. So this is not a key goal of environmental biotechnology. This is more aligned with industrial or food biotechnology. And these are some of the examples where plant and animal products are replaced by microbe, like big fermentation tanks where yeast is used for beer fermentation and cheese fermentation, and using genetically modified organisms and microbes to alter the soil availability, nutrients availability, and other industrial processes. So option A, as many of us answered, is the correct option. Replacing plant and animal products with microbial alternatives is not one of the key goals of environmental biotechnology. I hope it is clear to the students. We are having a detailed discussion on every question. Please let me know in the chat box if it is clear, and then we can move ahead. Very good. This is an interactive class, it seems like. Great. Now is the time for question number 3. Question number 3 is on display. Modern society faces many challenges which are interrelated, but the root cause can be traced to dash. Modern society has many challenges which are interrelated, but the root cause can be traced to dash. I'm sorry. Option A, mass extinction. Option B, energy shortage. Option C, overpopulation. Option D, food insecurity. And option D, disease outbreaks. Which of these is one of the major cause or one of the root cause for many of the problems faced in society of today or the modern society? Let us take a minute and collect some answers. I want everyone to participate in this class. Please post your answers in the chat box. I will be following them routinely. Okay. Which of these is the root cause for many challenges faced in the modern society? Is it mass extinction? Is it energy shortage, overpopulation, or food insecurity, disease outbreaks? These are 5 options. I hope everyone can read the question from the screen, as well as I'm reading it aloud for everyone. Okay, time to check the comments for answer box. Now, okay, there are already many answers. Nandini, Vidhi, Ankita, option C. Vishwa, Saida, Tapsi, option C. Mosmi, Manal, Snehal, Utkarsh, Yashashvi, everyone is answering option C. Okay, can we have some more answers? How many people are we there? I think 15 people. 15 or 14 people altogether. Okay. Let me see who are these students. Sorry if I'm pronouncing anyone's name wrong, but I will try my best. Okay, time to reveal the correct answer to this question. Modern society faces many challenges which are interrelated, but the root cause can be traced to overpopulation. Option C is the correct answer. Well done, everyone who have answered option C. Everyone has answered option C in unison. Overpopulation is one of the root causes for many challenges which modern society faces. And how is it? What is the definition of overpopulation? Overpopulation, or the state of having a population so large it strains available resources and ecosystems and has significant negative impacts on the environment, economy, and society. So overpopulation is one of the biggest problems even our country India deals with, right? Right now we are the highest most populated country in the whole world. We have surpassed China recently. So this is a big problem. And overpopulation impacts include increased resource depletion, environmental degradation, and social problems. So you imagine, assuming that all of you are biotechnology students, you imagine a bacterial culture which we do regularly in our research labs. If you grow bacteria in a designated culture volume, so we have the exponential growth curve. I'm going a little bit overboard, a little complex, but this is the perfect example for overpopulation. I hope you guys have seen the exponential growth curve for a population. So it happens with every population, right? If you remember, the exponential growth curves hit what is known as a plateau or a flat stage in which is called the lag phase. Log phase, which is the exponential growth, and then there is a saturation. And this happens when the resources of the environment are limited. So if you remember in your NCRT days, in your class 11th and 12th, you must have read it. What is known as the carrying capacity of a environment or a food patch. What is this carrying capacity? The capacity of an environment to support limited number of individuals. I mean, if I have food for 100 people, I can only support 100 people as an environment, right? Now, when population hits above this is known as overpopulation. Now, what will happen? There is competition for resources, there is resource depletion, there is less space to accommodate everyone, so there is environmental degradation and societal problems because there are now more people which will have unwanted interactions. So, that is what overpopulation poses a threat in the modern society. Now, how huge of an impact does overpopulation has? Overpopulation impacts the environment. Overuse of non-renewable natural resources. Non-renewable natural resources here, like petroleum, fossil fuels, right? Overuse of these, use of these are increasing because we have overpopulation. Habitat destruction and loss of biodiversity. Very important. Habitat destruction. A common example is mowing down of forest for expansion of land, for including converting more forest land into agricultural land, for buildings, for offices, and for commercial use. Loss of biodiversity, animals driven to extinction. These are all impacts of overpopulation. Pressure on land and soil degradation, leaching of soil, treatment of harsh chemicals. Overpopulation demands increase in food supplies, which increase in fertilizer. So it's all a chain which is affecting the environment very badly. Impact on economy. There is a strain in infrastructure because the same economy cannot sustain now an increased number of individuals. Widespread poverty and unemployment, as we can evidently see. More the population, the wealth is distributed between many people now, and there is higher chances of poverty and unemployment, not enough jobs to support increasing population, inequitable distribution, and the most important societal impact, lack of education to large populations and social unrest due to resource constraints, and healthcare also becomes poor. So these are some of the major challenges that are faced in the modern society today, and overpopulation is one of the root causes. I'm happy that all of you guys have understood this question and answered it correctly. Very good. Is it clear to the students? I think or simple these are simple and easy concepts, but anyways, as an instructor, I will explain everything in detail so that no one misses any detail. And while you read through your lectures, it will be easier for you to grasp these concepts. Is it clear to the students? Very good. Now coming to the fourth question on your screen. Fourth question says, earliest studies on environmental biotechnology were concerned mainly with dashed. Which of these kind of studies were first were the first kind of studies to be included in environmental biotechnology. Option A, wastewater treatment. Option B, enhanced oil recovery. Option C, valorization of waste. Option D, bioenergy. Option E, synthetic biology. I think this is an easy question for you guys. You guys seem very talented. This will be a piece of cake for you guys. Which of these is one of the earliest studies on environmental biotechnology? Is it wastewater treatment, oil recovery, valorization of waste, bioenergy, or synthetic biology? I think some of these people are joining very recently. Welcome to this class, and do not forget to join me every Saturday, 3 p.m. to 5 p.m. 2 hour session. We will fairly finish before 2 hours. A set of 10 to 15 questions in which we are solving multiple choice questions related to the corresponding week, and since it is the first week, we are dealing with week 1 of these lectures, which is the general introduction to biotechnology, dealing with all the definitions and the early experiments, the history of environmental biotechnology. Okay, let's not waste any further time, and let me check the comments for the answers. Okay, people have answered. Nandini, Vishwa, Saida, Snehal, option A. Almost everyone has answered option A. Utkarsh, Olivia, Tapsi, Tuhin, Manal, Vidhi, Ankita, Akshat, Mosmi, Mosumi. Okay, there is a question in the chat box. What does synthetic biology deals with? Okay, if you remember the first question, we deal with some of these examples of synthetic biology, which we will also come in the question ahead. But let me give you an example. Synthetic biology, synthesis of biopolymers and medicinal uses, in which we use enzymes isolated from microbes. The best example is enzymes like trypsin, enzymes like pepsin, which are isolated from bacteria and isolated from microbes in a large scale and used for synthetic applications like manufacture of medicines, manufacturing of polymers, manufacturing of galvanization, and many other industrial processes. So using the enzymes, so environmental biotechnology, if you remember the definition of environmental biotechnology, it can be either the use of the organism or any other component of the organism to drive the betterment of the environment, right? For synthetic biology, enzymes and many microbes are used for synthesizing, as I just said, synthesizing paints, for perfumery, for medicines mostly, for pesticides and agriculture. I hope that answers your questions. Nandini was asking the question. Priscilla has also answered recently, option A. Good. Now let us check the correct answer to this question. The correct answer is what the students have answered, option A, wastewater treatment. Wastewater treatment is one of the earliest studies which gave rise to this whole new field of environmental biotechnology. Okay, so let us see what is wastewater treatment. The earliest studies involved in environmental biotechnology were indeed focused on wastewater treatment, specifically the development of systems like activated sludge and anaerobic digestion of biosolids in the early 20th century marked the beginning of this field. So these are the two prominent examples of one of the examples which I have mentioned in the slide, right? Activated, the system of activated sludge and anaerobic digestion as well. These systems utilize microorganisms to break down organic matter in wastewater, paving the way for further advancement in environmental biotechnology. So, treating the sewage wastewater, breaking down the organic matter dissolved in the wastewater, separating the inorganic matter, and purifying the wastewater sewage treatment to be flown back into the rivers. This was the first idea where environmental biotechnology, where the use of microbes was indeed cultured. So this is a activated sludge process. In the activated sludge process, microbes are mixed with the wastewater. Microbes come in contact with the biodegradable materials or organic materials and consume them as food. We know many bacteria which are aerobic and anaerobic bacteria which consume these dissolved organic wastes in the water and turn them into biogas. In addition, the bacteria develop a sticky layer of slime around the cell wall that enables them to clump together to form biosolids. So the biosolids are nothing but aggregates of the organic matter along with the bacteria which have digested them as food, forming a slimy material on their cell wall and precipitating as biosolids. So this makes the mixed sewage water or slurry into two phases, separates it into two phases. One we see here in the secondary clarifier, one which is the sludge and which is recycled as the activated sludge which contains the bacteria in the first place, and biosolids which deposit as waste material. So these biosolids are sludge that is then separated from the liquid phase. Now the liquid phase can be released into the water body or as an effluent. So this is the process of active sludge, while this activated sludge, which is now the part of the biosolid, can be used together again for capturing the organic waste inside the water body. And this is another anaerobic digestion system, which is a well-known system for biogas production, where waste like agricultural waste, food waste, and waste water or the sewage sludge is fed into an anaerobic digester. In an anaerobic digester, we have anaerobic bacteria, and there is lack of oxygen. In the absence of oxygen, these bacteria convert organic matter into methane gas, mostly methane and butane gas, which is now released as a biogas, can be used as a fuel or electricity, and the solids that form can be deposited as biosolids. So these are two examples of the most primitive, how the most primitive wastewater treatment led to development of systems like activated sludge and anaerobic digestion, which boomed the field of environmental biotechnology. I hope everyone is clear. I think someone had some question. Can I say that synthetic biology is industrial biotech only? Yes, Nandini. Yes, I think you can. So industrial biotechnology, so we can say that synthetic biology is a subset of industrial biotechnology, because in industrial biotechnology we deal with many other processes, but synthetic biotechnology, synthesizing something synthetic like plastic or perfumes or medicines, these can be a subset of industrial biotechnology. So industrial biotechnology also refers to processes like fermentation in large cans for liquor, for beer and cheese, which does not come under synthetic biology. So synthetic biology is a subset of industrial biotechnology. That is what you can say. I hope it is clear to everyone. If you have any doubt, guys, you can post your comments or questions in the chat box. After the explanation, I will take a look and we will solve it and move forward together. Okay, moving on to the fifth question, since we have a lot of time left. Which of the following options shows the correct order of organizational hierarchy? Reading the question again. Which of the following options shows correct order of organizational hierarchy? Option A, population, organisms, communities, cells. Option B, cells, organisms, communities, population. Option C, genes, organs, cells, communities. Option D, genes, cells, organisms. Option E, organs, tissues, organ system, organisms. Come on. Basic biology. If you have a grasp of basic understanding of biology, you will be able to answer this question correctly. there won't be any issues. What is the correct order of organizational hierarchy? Let us take a minute and collect some answers. Write your answers in the chat box. You can write the full answer or just mention the option A, B, C, D. I'll be checking the chat box. Wow, immediately there are so many answers. Me, within reading the question completely, there are already answers now. Okay, Vidhi, Saida, Snehal, option D. Nandini, Olivia, option B. Vishwa, Utkarsh, option D again. Akshat, Tuhin, Ankita, option D. Okay, I don't think we have to wait any longer on this question. Any more answers? People who didn't get a chance to answer these questions? Which of the following options shows the correct order of organizational hierarchy? Come on. And people who do not have a stable internet connection, please try to rejoin this call. I will be admitting everyone who tries to rejoin this call, and pass on this link to your friends who want to attend this class. Does not matter if they have registered for this course, they can just come in, interact, and gain knowledge from these classes, right? Okay, time to reveal the correct answer to this question. Correct answer to question 5 is option D. This is the correct organizational hierarchy: genes, cells, organs, and organisms. Genes, most miniature of the hierarchy. Then comes cells. Genes are within the cells. Then comes organs. Cells within many cells make up organs, and then organisms. This is one of the only correct order of organizational hierarchy. Is there a comment? Manal says option B. Sorry, Manal, you were not correct. Vidhi, okay, Vidhi was changing her option, but yes, Vidhi, you were correct on the first time. D is the correct. Yeah, the symbol is just an arrow mark here, showing it's not the greater than symbol actually. It's showing the sequence, like the smallest to the largest, any kind of organization. Population, organisms, community, cells. Even option A is not the correct answer, because communities will come within populations, right? Even if the sequence was correct, population, it would be then communities, then organisms, and then cells. Okay, let us look at the biological hierarchy of organisms. So this is the best slide to explain the organizational level in our environment. So the largest, we are moving from the topmost organization level to the smallest. Okay, come with me, let's follow it from the top to the bottom. At the topmost level is what is known as the biosphere, the part of earth that contains all ecosystems. So this is known as biosphere. It literally translates to sphere which holds life, bio and sphere. So all the parts of earth that contains all the ecosystems, the terrestrial ecosystem, the ocean ecosystem, the aerial ecosystem, everything, right? Now what is an ecosystem? Community and its non-living surroundings. So ecosystem consists of community of organisms, both living and the non-living surroundings of the organisms. So this constitute an ecosystem. Not as large as the biosphere, but many ecosystems constitute the biosphere. Moving one level down is the community, populations that live together in a defined area. Now those become community. So see, option A would have been a correct answer if communities would have come before populations. Sorry, I had made a small mistake. Communities, population, organisms, and cells. Okay, that would have been the correct option A. So communities are populations that live together in defined areas, like the Indian community. Populations of Indians living in the Indian subcontinent is the Indian community. Now, what is population? Group of organisms of one type that live in the same area, like a population of bison, a herd of bison, a herd of elephants, a pack of lions, and a pack of wolves. All these are populations. And then many such population interact to give rise to a community, and community interacts with its non-living components to give rise to ecosystem. Now, organism, which is one level down from the population system, a single individual living being is an organism. Now, inside the organism, we come to the organ system level of organization, which consists of tissues, organs, and organ systems like the nervous system, the circulatory system, the skeletal system. All these systems make up the organism as a human being. Then comes the cells, which are the smallest functional unit of life. Like a nerve cell is the smallest functional unit of a nervous system. And then comes molecules, which are inside the cell, which are the macromolecules, which are building blocks of the cells, such as proteins, carbohydrates, the genes, the DNA, ribonucleic acids, the proteins, the lipids, and the carbohydrates. So this is the organization in biology, biological and environmental organization, in which we start from biosphere and end up with the smallest cells and molecules. So the organizational hierarchy in biology represents the level of organization from the smallest to the largest units of life. So you need to remember this definition. Anywhere you get asked the question about organizational hierarchy, it's always from the smallest to the largest units of life. So option D is the correct answer. Genes are basic units of heredity made up of DNA, smallest level of organization. As we just saw in the previous image, genes are found within the cells, which are capable of fundamental building blocks of life, capable of performing all life processes. And then these cells come together to make up organs. Organs function, carry out specific function, and organs make together an organisms. So this is the correct order of biological hierarchy, and option D is the correct answer. I hope it is clear to the students, and it is simple and clear. If you just remember this slide, that is enough for you guys to memorize the correct order of biological organization. I hope it is clear to the students. Very good. Thanks for responding in the chat box. Let us move to the question number 6. Question number 6 on your screen. With respect to a pond ecosystem, identify the correct option about primary producers. Which are the primary producers in a pond ecosystem? Now you guys have to memorize or recall what is a pond ecosystem, what are the living components of a pond ecosystem, what are the non-living components, and which of them are producers. Primary producers, not just any producers, primary producers. Primary producers in the sense the main source of producers. So I'll read out the options for you guys, but I don't think I need to, because people have already mentioned their answer in the chat box. Aquatic plants and algae, option A. Option B, insects, fish, etc. Option C, anaerobic bacteria. Option D, methanogenic archaea. Which of these are the primary producers in a pond ecosystem? Option A, is it option B, C, or D? Let us take a minute and let everyone answer present in the class. Please mention your answer in the chat box, as well as people who will be watching this recorded video. Let us take some time for them. Although I already see immediate answers while I post the question on the slide. Nandini, Snehal, Olivia, Vishwa, Utkarsh have all answered option A. Vidhi, Krishanu, Tuhin, Ankita, Saida, Manal, and Mausumi. Everyone has answered option A. And I'm very happy to see that people are participating in this class. It's good and enthusiastic for a instructor or a teacher as well. So the correct answer to this question number 6, is none other than option A, aquatic plants and algae. Congratulations to everyone who has answered this correctly. Everyone almost has answered option A to this question. Aquatic plants and algae are the primary producers in a pond ecosystem. Okay, now let us have a look at what how does a pond ecosystem look like, right? Pond ecosystem is a self-contained freshwater environment where various living organisms and non-living components interact and depend each other for survival. So basically it's a pond, or in Hindi we say pokhar, right? I hope everyone, I know the official language is English. I will try mostly to communicate in English, as this will be a recording and will also be accessed by and understood by anyone in the future. Okay, so the pond ecosystem is a is a small ecosystem in itself. And why is it called an ecosystem? Because it has both living and non-living components interacting with each other, and it's a closed space and system. Now, what are the key aspects of the pond ecosystem? The biotic factors. Biotic factors or the living factors include, like, these are the producers again: aquatic plants and algae. Aquatic plants like lily, lotus, flowers, and seagrass, seaweeds. Consumers like many kinds of insects, aquatic insects. Zooplanktons. Many a times we confuse zooplanktons as the producers in aquatic ecosystems, but it is not. They are not the primary producers. Zooplankton actually feed on phytoplankton. So phytoplanktons are the producers, but zooplanktons are the consumers. Then obviously there are fish and amphibians and reptiles and decomposers like bacteria and fungi. So all these components make a pond ecosystem. There are biotic factors, and biotic factors includes producers, consumers, and decomposers. So any ecosystem, any environment which has all these components can be declared an ecosystem. So there are fungus and bacteria and other detritivores which act like decomposers in the pond ecosystem. Now, what are the abiotic factors? The non-living components such as water, sunlight, temperature, nutrients, and physical structure of the pond, like the soil inside the pond, the mud, the sand on the pond banks, and the soil on which the aquatic plants grow in. So here is a beautiful representation of a pond ecosystem on my slide, mentioning the biotic factors: producers like algae and hydrophytic plants, consumers like protozoa and crustacea, like crabs and fishes, insects and larvae, sharks. Not many sharks in the pond ecosystem as well, but this is an exaggeration. Decomposers like bacteria, fungus, and microbes. And the abiotic factor or the non-living factors like light, heat, and water. Sunlight which shines on the water surface. Heat and water and minerals present in the soil, dissolved gases dissolved in the water, are all the abiotic components. Now, what kind of interactions happen in a pond ecosystem? Organisms within the pond ecosystem interacts through food chains and food webs. Like, producers are the algae, which are consumed by insects, and zooplankton and fish, which are consumed by the larger fish, which are then dead and decayed by the decomposers. Producers forming the base and decomposers recycling the nutrients and supplying it with the producers, like any other food chain. And ponds are important habitats for various species, including endangered species, and contribute to overall freshwater diversity. Let us have an example of the pond food chain. The algae, being the primary producers, is eaten by the small fish, which is then eaten by the big fish, and heron is the final consumer or the tertiary consumer, the apex predator, which eats the big fish. And the remains of this fish is now decomposed by the detritivores present in the pond ecosystem. Once again, producers in the pond ecosystem are primarily algae and aquatic plants. These organisms, through photosynthesis, convert sunlight into energy rich compounds that form the base of the pond's food chain. So, without any algae, there won't be any pond ecosystem because these are the energy provider in the pond. They fix the sunlight for energy via photosynthesis and provide the base of the food chain. They provide oxygen and food for other organisms in the pond. So, as everyone has answered very correctly, primary producers in the pond ecosystem are algae and aquatic plants. I hope it is clear to the students. Just let me know in the chat box and we will move forward. Very good. It's good that you guys are following and you guys are with me, paying your full attention. For students who are joining very recently, uh, my name is Ashutosh Behera and I'm a PhD scholar at IIT Kanpur, and I'll be your teaching assistant for this course. We'll be having the live sessions every Saturday 3 to 5 pm unless prior notice, in which we may shift classes. And these are all recorded sessions where we will solve exemplar questions, which is very similar to the assignments for people who have registered for this course and certification of this course. These are pretty easy questions which will help you to recap the lectures as well as help you solve these assignments. You can post your doubts, ask your questions in the chat box, interact. Let's keep this session interactive. Okay, let us move to the next question. Question number 7. Which of the following statements are true with respect to the Gaia hypothesis? Now guys, if you remember the Gaia hypothesis, I hope you do. Which of the following statements are true with respect to the Gaia hypothesis? The Earth is a single sentient superorganism. The Earth is a single sentient superorganism. The Earth seeks to apply selective pressure to enhance growth of specific organisms. The Earth's abiotic systems behave as a single independent organisms. The Earth is attempting to return to a homeostatic state by eliminating all organisms, and the Earth's biotic and abiotic systems interact to preserve life and maintain self-regulation. Now you have to read the question carefully. Which of the following statements are true with respect to the Gaia hypothesis? Only one of these options are correct. The Earth is a single sentient superorganism. I have mentioned all the 5 options. Please take your time and let me know what you think is the correct answer. Post your answers in the chat box. I already see many answers. I'm sorry, there was some problem, I think. Let me reshare my screen. Don't know what. Can everyone see my screen again? There was some issue and the presentation was not visible, I think. Can everyone see my screen again? Yes, okay. Okay, I see some answers already. Nandini says A, Snehal A, Vishwa A, Olivia is confused between A or E, Mausumi E, Manal, Utkarsh E. Okay. Any more answers? People who didn't get a chance to answer, please come forward. We are at the 7th question. I think I have prepared 10 questions for you guys, so few more to go and we will be done with this class. So I want everyone to participate. Please answer. Very easy question if you remember the Gaia hypothesis and the prominent scientist who had proposed this hypothesis, you will be able to answer this. Okay, I think no more answers. Let me reveal what is the correct answer to this question. Which of the following statements are true with respect to the Gaia hypothesis? And the correct answer is option E. Option E is the correct answer, guys. The Earth's biotic and abiotic systems interact to preserve life and maintain self regulation. Right? Option E is the correct answer. Now let us understand what is the Gaia hypothesis. The Gaia hypothesis was proposed by James Lovelock. Okay, someone has answered. Manal says proposed by James Lovelock. Very correct. Well done, Manal. You have answered very correctly. James Lovelock is a scientist who proposed this idea that the whole Earth, all living things on Earth, which is the biosphere, function as one super organism that changes its environment to create conditions that best meet its needs, with the ability to self-regulate critical systems needed to sustain life. So in a sense, option A also fits into this answer of Gaia hypothesis. The Earth is a single sentient superorganism, but if you dissect out the components, it is not the Earth, but the components inside the Earth, both the biotic and the abiotic components, which interact and then make the biosphere as a sentient organism. So option E fits more to this answer. I think everyone would agree with me. Olivia, Mausumi, Manal and Utkarsh have answered option E. Those who have answered option A, E is a better fitting answer to this question. That is why E is the correct answer. Yeah, so this is the Gaia hypothesis. All the living things function as one or superorganism which changes its environment to create condition that best suiting itself. So any living being, I think many people are trying to join this call. Maybe net is an issue and they're trying to rejoin this call, because we are a few group of students, I think 17 or 18 students. Okay, let us coming back to Gaia hypothesis. The ability to self-regulate the critical systems. All these living things have the ability to create an environment which suits themselves or sustains life on Earth. This is the Gaia hypothesis proposed by James Lovelock, and the best or most cited example of the Gaia hypothesis is the carbon dioxide cycle. In the carbon dioxide cycle, what happens in the CO2 cycle? Sunlight is fixed. Sunlight, in the presence of sunlight, plants or the organisms, living matter, fix carbon dioxide in the process of photosynthesis, and then when they decay, they provide organic carbon into the soil. The dead and decaying matter provides organic carbon into the soil, and these now collect in the pool of dead organisms and waste products, while other animals which perform exhale CO2 in respiration, exhale CO2 in the atmosphere, which is then fixed into the plants by photosynthesis. This CO2 carbon reserve undergoes fossilization and fossil fuels, and this is also taken up by plant with plant respiration and then released back into the atmosphere. So this cycle from the atmosphere inside the soil through these living organisms and recycling of carbon dioxide is called the CO2 cycle. The CO2 cycle involves processes like photosynthesis, respiration is a prime example of how living organisms regulate atmospheric composition and temperature, contributing to the biosphere's homeostasis or Earth's homeostasis. So this is one perfect example of the Gaia hypothesis, the CO2 cycle. Water cycle is another good example of the Gaia hypothesis, given by Lovelock. Is it clear to the students? I hope everyone would now be able to remember the Gaia hypothesis. Let me know in the chat box, and we will move forward to the next question. Very good. I like a responsive class, and good to know that people are interactive. Okay, moving on to the next question. Question number 8. Nutrients in soil are made accessible to plants by the activity of dash. Nutrients in soil are made accessible to plants by the activity of dashed. Option A, producers. Option B, primary consumers. Option C, secondary consumers. Option D, detritivores. Option E, autotrophs. Now, which of these compositions, which of these components of the food chain, these are all components of the food chain and part of the ecosystem. Their major role is to provide nutrients back to the soil, availability of nutrients in the soil. Are they producers? And these nutrients is made accessible to the plants as well. Do not forget this point. Are they producers? Are they primary consumers? Secondary consumers, detritivores or autotrophs? Let us take a minute and collect some answers. And excuse me for a while, I will go grab my water bottle to drink some water. Meanwhile, keep posting your answers in the chat box. I will follow the answers shortly. Okay, I think it's time to check the comments for the answers. There are so many answers. Wow. Nandini, Vidhi, Vishwa, Utkars, Sai, option D, Snehal, Moshami, Tuhin, Manal, Saida, Olivia, Ankita, everyone have answered. Subhashmita, Bhagyalakshmi, Krishano. Option D, okay. Now if everyone is answering option D, there is no chance that this cannot be the correct answer. Option D is the correct answer. Detritivores are those components of the ecosystem which are responsible for making the nutrients available in the soil for plants to absorb. That is the role of detritivores. Now, what are detritivores? A detritivore is a heterotrophic organism. It is not an autotroph. Autotrophs are the ones which can make food themselves, which fix the energy of the sun in the process of photosynthesis and create energy for themselves. A heterotroph is one which depends on other organisms for its nutrition and energy. Basically, a heterotroph depends on an autotroph for its nutrition. A detritivore is a heterotrophic organism that obtains its nutrition by feeding on detritus. And what is detritus? Detritus is the organic matter made up of dead plant and animal material. Detritivores may also obtain nutrition by coprophagy, which is a feeding strategy involving consumption of feces. So there are many detritivores which can also feed on feces or the fecal material, and this is known as coprophagy. So we learnt two different terms today: detritivore, which feeds on detritus, which is the dead and decaying matter or organic matter, and coprophagy, which is the consumption of feces, which is also carried out by detritivores. Detritivores are often invertebrate insects such as mites, butterflies, beetles, flies, mollusks such as slugs and snails, earthworms, millipedes, and woodlice, termites. These detritivores eat dead organisms, dead wood, and other dead material. So the termites which eat our wooden doors and furniture are none other than the detritivore, because we build our furnitures with the dead and decaying wood. Then earthworms, one of the major contributors to soil enrichment and nutrient enrichment, very important organisms for agriculture and very keystone organisms for any ecosystem. Crabs and flies are also examples of detritivores. Now here is a cycle showing the importance of detritivores and how the nutrient cycle is maintained via the action of these detritivores. Detritivores like earthworms, millipedes, and insects physically break down detritus into small pieces, increasing the subsurface area for microbial decomposition by bacterial and fungi. So decomposers and detritivores are different things. Detritivores are a subset of decomposers. The actual decomposers are microbes like bacteria and fungi. Detritivores actually chop down the organic matter into smaller pieces, increasing surface area and absorption and decomposition by these microbes. And they also enhance nutrient release. Through their feeding and excretion, detritivores release nutrients like carbon, nitrogen, phosphorus, calcium, and potassium back into the soil or water. This process of decomposition and nutrient release is vital for nutrient cycling, ensuring that essential elements are available for plant growth and other organisms. So here is a diagram representation showing how carbon dioxide or carbon and nitrogen from the atmosphere are fixed into the plant. Whether if it is a leguminous plant like which harbors rhizobium bacteria, symbiotic fixation of atmospheric nitrogen or fixation of carbon through photosynthesis, then organisms which die off, and these plants which die off, they are eaten up by detritivores like earthworms, mites, and springtails, which then feed it into the decomposers comprising of bacteria and fungus like aerobic and anaerobic bacterial and fungi. Many plants also harbor fungus in symbiotic relationship and other microbiota which help in decomposition and mineralization, fixing of minerals like nitrogen, phosphorus, and potassium, and enriching the soil for the plant to absorption. So this is the nutrient cycle which is brought into place by important components of the nutrient cycle called detritivores. Like main examples include earthworms and termites and millipedes. I hope it is clear to the students. I'm very happy that everyone has answered very correctly. Option D, detritivores is the answer, which are the nutrient cyclers of the ecosystem. Very good. Thanks for letting me know in the chat box. Moving on to the ninth question of this session. Prokaryotic microorganisms are the most genetically diverse organisms on Earth. Which of these is not a probable explanation for this trait? Understand this question. Prokaryotic microorganisms are the most genetically diverse organisms on Earth. Which of these is not a probable explanation for this trait? Option A, they reproduce quicker and thus evolve quicker. Option B, they are small and can easily evade most predators, persisting longer in the environment. Option C, they were the first life forms to evolve, hence they had been evolving for a longer time. Option D, the DNA replication machinery is in general less robust and tends to create more mutations. Option E, they can perform horizontal gene transfer relatively easy. Mind the question, which of these is not a probable explanation? Which of these is not an explanation for microbes having such genetic diversity, for prokaryotic microorganisms? Take your time to understand the question and post your answer in the chat box. We will revisit this question in a minute or so. Come on, I'm checking the chat box. Everyone, please post your answers. Everyone present in the class, don't shy out. I'm not asking you to turn on your video or speak up. You can silently post your answers in the chat box and still be interactive, right? Don't shy up. These sessions are meant to be interactive. That is why NPTEL has organized these live sessions and asked PhDs like me to teach and train you on this subject. Okay, I see many answers in the chat box, but there are mixed answers. Vidhi says D, Nandini says B, Utkarsh B, Olivia said D, Snehal B, Ankita D, Vishwa says D, Olivia has changed to B, Tuhin and Tapsi are saying B. Okay, a mixed response between option B and option D. I think people are getting confused. Okay, time to reveal. Okay, there is another answer. Manal thinks it's A. Okay, thanks for answering, but let me reveal what is the correct answer to this question. Correct answer to this question is option B. They are small and can easily evade most predators, persisting longer in the environment. This is probably the not this is probably the wrong explanation for this trait of microbe. Understand this question carefully. The question is talking about genetic diversity of prokaryotic microbes and asks us which of this option is not a probable explanation. So it's not true that microbes can evade most predators and persisting longer in the environment. No. Microbes are one of the most consumed organisms, right? Microbes are generally eaten by larger organisms as prey, and persisting longer in the environment due to predator evasion is not the driving factor for genetic diversity. So you have to understand this, that this option fits the best for not being a probable explanation. Option A is a genetic cause for creating genetic diversity. If you have quicker reproduction times, you evolve faster. And option C, they were the first life forms to evolve. Yes, they were. And therefore, they have longer time for evolution. That is why they have a larger genetic diversity. And DNA replication machinery is designed as such that it incorporates mutation, creating genetic diversity. And they can also perform horizontal gene transfer. Horizontal gene transfer is something which is very specific to prokaryotic microorganisms, which is a specific quality which determines the genetic diversity in the microbes. Okay, so option B is the correct answer. Prokaryotes encompassing bacteria and archaea are the most genetically diverse domain of life due to combination of factors, including rapid reproduction rate, as we see in option A, large population sizes, and the prevalence of horizontal gene transfer. Okay, there is a question in the chat box, I think. Can you give some example for predators of prokaryotes except for larger prokaryotes, ones who directly consumes them? Okay, let me give you an example of a protozoan called Euglena. Euglena is a protozoan. Euglena feeds on prokaryotes like blue-green algae, and they directly consume these organisms. And there are other parasitic organisms which are eukaryotes, which directly consume prokaryotes. And one of the best example of consumption of prokaryotes via the eukaryotes is the endosymbiosis theory, in which mitochondria, which was an early prokaryote, could have been ingested by a larger eukaryote as a form of residing within them in symbiosis. So Euglena is one of these examples, and Paramecium is another example in which a eukaryote can directly feed on a prokaryote, because these feed on smaller prokaryotes. Great. Okay, coming back to the explanation of this question. Yeah, prokaryotes have these factors, including rapid reproduction rate, large population sizes, and prevalence of horizontal genes transfer. Here is a diagram showing all the methods of horizontal gene transfer by the process of conjugation, transduction, and transformation. Horizontal gene transfer means transferring one organism transferring gene to the other organism, and which is not its offspring. It can transfer it to a neighboring organism. Direct gene transfer or vertical gene transfer is gene transfer from a parent to an offspring, like humans. Human can transfer their genes from a parent to their child, but bacteria can transfer their genes from one neighbor to another, forming these kind of structures called pillars. These mechanisms allow for rapid spread of genetic material even between distantly related species. See, even between distantly related species, this allows prokaryotes for rapid spread of genetic material, leading to vast array of genetic combinations and adaptations. That is why it is one of the most genetically diverse organism. So looking at the characteristics in details, again there is a question in the chat box. What are gene transfer agents? Some examples. Okay, gene transferring agents. So if you're talking about gene transfers in bacteria, then there are certain extra chromosomal DNA. So the bacteria has two types of DNA. One is the chromosomal DNA, which is the genetic material of the bacteria, and the other, which is the extra chromosomal DNA called plasmid. Yes, you are correct. I think Vidhi and Nandini have helped each other in the comment section. It's a plasmid, right? So there are certain plasmids which give characteristic to a bacteria to form a pilus. These kind of plasmids are, some examples are fertilization plasmids or F plasmids. So some bacteria have these kind of F plasmids which can form an elongated bacterial structure, forming a tube through which it can transfer its genetic material to another bacteria. So yes, you have answered it correctly. Thanks Vidhi for helping Nandini out. Plasmids are an example of gene transfer agents. Yes, looking at the characteristics in more detail, higher reproduction rates of prokaryotes, particularly bacteria, double their population in matter of minutes. You know, as an example of E. coli doubles its population in 20 minutes. Every 20 minutes, there is a bout of reproduction of E. coli. One E. coli will turn into two. This rapid reproduction allows mutations and spread quickly within the population, increasing the genetic diversity of smaller organisms like prokaryote. Large population size. Prokaryotes often exist in large population size because of the smaller physical size, and this provides a greater chance for beneficial mutations to arise and to be maintained, as we see in antibiotic selection. When you select a group of bacteria, there are some bacteria which gets resistant to these antibiotics and develops genes for antibiotic resistance. So these are also examples of beneficial mutations and how these genes are now getting transferred across populations conferring antibiotic resistance to bacterial populations. Horizontal gene transfer, which we saw as an example, which is conjugation. Metabolic diversity, many bacteria, metabolic diversity and extremophiles, many bacteria have wide range of metabolic dependence. An astounding array of metabolic pathways allowing them to thrive in extreme conditions with wide range of energy sources. They are not restricted to one source of energy. They can utilize carbohydrates as source of energy, as lipids and proteins, and they can thrive in many extreme conditions. And these kind of bacteria are called extremophiles, such as radiation contaminated sites, highly saline environment like salt flats, hypersaline environment, acidic and alkaline environment, deep sea hydrothermal vents where there are high temperatures, and polar regions. So in such areas you will only find prokaryotics, bacteria and archaea. You will never find eukaryotic organisms, because bacteria have such diversity of molecular and metabolic pathways which allow them to thrive in many conditions. So yes, these factors allow the prokaryotes to achieve such a high genetic diversity. I hope it is clear to the students. If any other questions, what is the difference between extremophiles and archaea? So again, archaea are a kind of prokaryotes which were first thought to be bacteria, but they share distinct features from the bacteria. Extremophiles are a group of bacteria which can thrive in extreme conditions like hydrothermal vents, like hypersaline environments where the salt concentration is too high. You can find extremophiles, and sulfur containing bacteria in hydrothermal vents. These are the examples of extremophiles. While archaea are another ancient form of prokaryotes which differ from bacteria. Some archaea even lack a cell wall or a cell membrane, and most prokaryotes, they do not have a nucleus. They do have a genetic material though floating in a space called nucleoid. So they are very similar to bacteria, but now they are classified into a separate kingdom of classification called archaea. While archaea can also be extremophiles, any organism or any bacteria found in extreme conditions are extremophiles, while archaea is a different kingdom of classification from bacteria, and all of these are prokaryotes. I hope this clears your confusion. But aren't all archaea extremophiles? Yes, most of these archaeas have properties in their cells, cell walls and internal properties which makes it a clear statement that most of these extremophiles are archaea. You are correct, Nandini. But there are also other types of prokaryotes which are found in extremophiles, but mostly archaea. Any example of those who aren't? Okay, I right now do not remember any examples, but I will get your question in the next class. Any examples of archaea which are not extremophiles? Okay, I will note it down and clear it in the next class. Please remind me in the next class. Let's move on to the next question. Final question of this session. Majority of biotechnology process relevant to environmental applications make use of microorganisms because dashed. What is the primary reason why biotechnology process relevant to environmental application make use of microbes? Option A, microorganisms have very specific growth requirements. Option B, microorganisms can easily adapt to variable conditions. Option C, microbial biocatalysis is very expensive. Option D, microorganisms are quite small in size. Option E, microorganisms grow slowly. Why many biotechnology process for environmental applications make use of microorganisms? Post your answers in the comment box, and I will visit the question in a minute. Okay, I think there are many answers in the chat box. Which of the which of these is one of the major characteristics for why microbes are used for environmental applications? I think we dealt with a similar kind of question uh, before. Okay, so all the students have unanimously answered option B. Nandini, Snehal, Olivia, Vishwa, Yashasvi, Tapsi, Vidhi, Saida, Moshumi, Krishanu, Ankita, Tuhin, Manal. Olivia is asking, can D also be an option? Let us see what is the correct answer to this question. Option B is the correct answer. You guys have answered all correct. Well done. And Olivia, yes, D can be an option, but majorly for environmental applications, option B is the correct answer. Because they can adapt easily to various conditions, now they can be used for more environmental applications. For a small size, they can be used for industrial applications. Correct. Option D can also be an answer. Environmental biotechnology deals with cleaning up of pollution, treating waste, and restoring ecosystems. These environments can be very harsh or change frequently in temperature, pH, oxygen levels, presence of toxic chemicals. So that is why we need our microbe to adapt to every condition. Microorganisms are ideal for such tasks because they are highly adaptable. Many species can survive and function in changing or extreme environments, and they evolve quickly. Microbes can mutate and adjust their metabolism to degrade a wide variety of pollutants, and they are naturally present in soil and water, already doing much of this work. So they are highly adaptable. They are highly adaptable to many pH and toxic levels and oxygen levels, and they can evolve quickly to match their requirement. That is the major characteristic that make microbes the best use for environmental applications. And thus microorganisms are used for applications like agricultural uses, industrial applications, which was option D, environmental application, which was option B, and synthetic biology and pharmaceutical applications. Everywhere in every field, microbes have found their application because they are very adaptable. Okay, is it clear to the students? Great, very good. With this, we have come to the end of our class. Let me stop my presentation. Okay, with this, we have come to the end of this class. I hope this was an informative and interactive session for everyone who has joined this meeting. This is a recorded class, and as soon as I upload this, we finish this class, I will upload this on a YouTube link, and this will be made available to you through the NPTEL. And the slides that I have presented today will be shared to you through the NPTEL authorities. Thanks for joining this class, and if you have any feedback regarding the way this session is conducted, you can tell me if you like it or not, if you want any changes, if you want more questions, if you want lesser questions, if you want to change the way I explain. And thank you for joining. See you on next Saturday, where we will deal with lecture week 2. Please go through your lectures and be ready for the live session where we will solve some questions. Bye bye and enjoy your evening. Who will take the question I post? I think this is a comment from Nandini. Nandini, I will take your question if you join the next Saturday class. I think your question was, you were asking about any examples of archaea which were not, which were not extremophiles, right? Okay, Moshumi is helping you out. Brine shrimp, sea monkeys are the extremophiles and aren't archaea, and there are more in plant species. Okay, great. see, you already have a classmate solving a doubt for you, which I had to learn about. I didn't knew it. I couldn't remember the name, but definitely, if you have any more questions you want, and right now do not have the answers, I will take it up in the next class before beginning the next session. Okay, Nandini is asking something else. Is 20 what? But I posted one question during the lecture. I'm sorry, but I'm not aware if you what happened during the lecture. What what we are dealing with the live session here is some of the example questions. But if there was a live lecture from Professor Pinaki and you have posted it there, then I do not have any idea about the forum. Okay, recorded sessions. Oh, I think this is the first session that we are going to have. We are having, I guess, because from 25th July onwards, we were going to have the live session. And if you had posted in the recorded session, then it must be some other teaching assistant or an instructor. The sessions professor took. Okay. Okay, let me see. I will definitely check if you have any question in the recorded section in the forum. I have to access the SWAM portal, and I will get back to you in the next class because right now I have no idea where is this forum thing you're talking about. There was an option of asking questions, so I did. Yeah, great. You should definitely ask questions. If you can narrate the same question to me in the next class, I will be able to help you out. I don't know if it is related to lecture week 1 or 2, because as a teaching assistant I have to read the course as well while preparing for these slides and while preparing for the class. So if you can let me know. Yeah. And Moshumi, thanks for helping us out. Thanks for answering Nandini's question. There was another question from Olivia, I think. Is 29th a different Q session? 29th. So my question answer live session will be on the next Saturday, which is I think second of August, or I don't remember. Maybe 2nd of August. Yeah, 2nd of August, 3 to 5 p.m. will be my live session. If there is another live session which I do not know of, you're free to attend. But I don't know if there is a 29th question session. I have been allotted this slot for taking up assignment. I think if you find it informative, please join this class and ask your friends if they can gain any knowledge. Okay, I think we have reached almost reaching the end of this session, and thank you for joining. I will end this call now. See you soon on the next Saturday. Thank you for joining.